Let us look at the different processes that happen during the gastrulation process of fish development. Now let us now look at the various cell movements that happen during the blastula stage. So during the blastula stage itself, cell movements will begin. Now the first cell movements is the epibole of the blastoderm cells. Now the deep cells in the blastoderm will intercalate with cells that are closer to the surface. And when we look at the figure to the left, we are seeing where epibole is right about to begin. And pay attention to the part in the blue box, which is the upper part, and that is where the blastoderm is. Now, we can now observe the cell movements happening where epibole is beginning, and we can see that there is a thinning of that blastoderm. So it is thinner than what we see in the figure to the left. This is because of the intercalation of the inner deep cells with the deep cells that are closer to the surface. Now the yolk cell, which has the syncytial nuclei, pushes upward to form a dome shape. And so if we again focus on the figure to the left and look at where the part of the yolk cell meets the blastoderm, then we can see that when epibole begins, there is a change in its shape and it looks more dome-shaped as the cells of the blastoderm start thinning. Now the movement of yolk nuclei results in the internal Y cell that is right below the blastoderm, which we saw in the previous slide, and the external Y cell that is in the upper cortex. This is what is shown in this figure where we can see the inner Y cell shown in the blue box. And now I'm going to change the location of the blocks, and this is where we can actually see the external Y cell being present. All the green cells that are forming a rainbow-like structure, those are the deep cells that are present. Now, as the blastomeres undergo epibole to cover the yolk cell, I already mentioned that they tend to thin out through intercalations, and this is through the radial intercalations. So, when we look at a portion of the embryo before epibole begins, we can see the blastomeres and the EVL section. Now, once epibole begins, we can actually see how the intercalation occurs. The red cells are the cells that are the deep cells that are inside or in the inner part, whereas the orange cells are deep cells that are closer to the periphery. And once we have the radial intercalation, we can actually see the mixing of the red cells with the orange cells, and now we can see that that layer is not as thick anymore. Now at 30% epibole, and in the case of zebrafish, the progress of the cell movements is denoted in terms of a percentage of epibole. A 30% epibole basically means that the percent of the yolk cell that the blastoderm covers is 30%. And in this stage, the EVL is 1 cell layer thick, while the deep cells tend to be about 4 cell layers thick. And thus, we can now already see that the EVL has become one sheet, and the deep cells have started becoming thinner. Now, around 50% epibole, that is when gastrulation will begin. During the process of gastrulation, involution occurs. And the involution generates a thick band at the marginal region of the blastoderm rim, which is called as a germ ring. So if we look at the figure A and compare that with figure B, we can see a bulging portion where there is the blue box, and that is where the germ ring is. Now in fish development, the blastocele and archenteron are absent. So if we compare this with amphibian development, we saw the presence of a blastocele, which was that fluid-filled cavity that is surrounded by cells, and we don't see that in the case of fish embryogenesis. We also saw the formation of an archenteron, and a blastopore, and hence in the case of fish, archenteron once again are not developed. Hence, for a blastopore to form, the deep cells undergo involution. Now, the germ ring is formed by involution of blastomeres, and this involution of the blastomeres will actually form two layers: the outer epiblast that will eventually form the ectoderm, and the inner hypoblast that will form the mesoderm and the endoderm. Now, if we look at the figures, then the figure in the blue box is actually showing the process of involution, where the blastomeres shown in green are coming down to cover the yolk cell, but in the process, involution also occurs where we can see those brown cells that are being pushed upward to form a second layer. When we look at a magnified image, we can see that the green cells are the epiblast, whereas the dark orange cells are the hypoblast. And the C-shaped cells or green that are the outermost cells are the enveloping layer. Thus, the process of involution ends up forming two layers, the epiblast and the hypoblast. Now, when we look at the final figure, that is the process that is actually showing the process of involution. Now, in this case, the epiblast is shown in blue color. Now, those cells are actually going downwards to cover the yolk cell. But as they're going downwards, some of them are slid upwards also to form the inner hypoblast that is shown in red. The process of epiboly temporarily halts when the germ ring is formed. However, after the formation of a structure called as the embryonic shield, epiboly can continue. Now let us see how the embryonic shield is formed. So when we look at the germ ring, the cells at one location will accumulate, and they tend to accumulate at the dorsal part of the germ ring. And this structure is called as the embryonic shield. So when we look at the figure in the blue box, we're looking at the embryo where the germ ring has formed, and hence there is an arrest in epiboly. In the next figure, when we look at the part of the blue box, that is where we can see the accumulation of cells in the dorsal part, and this is the embryonic shield. Now, when we look at the part that's in the blue box, this is where the blastoderm has undergone through epiboly, and once the embryonic shield has formed, the epiboly process will continue. And we can see in the last figure in the blue box, that is where now we can see that part of the yolk has been covered through the process of epiboly. And so in the case of the panel F, most of the yolk cell is actually being covered by the epiblast. Now the embryonic shield is similar to the dorsal blastopore lip of amphibians in terms of its function, and that it acts as an organizer of the fish. Now, during the development process, cells converge at the embryonic shield and they extend anteriorly. And this is what is shown in the figure in the blue box, where we can see that cells move downwards to cover the yolk cell, but through the process of epiboly, we are also seeing cells being rolled under, which is forming the hypoblast, and those cells will undergo convergence, and they tend to extend upwards towards the anterior region. Eventually, this strip will narrow down along the dorsal line, and that is what is shown here, where you have the shield structure, and if we follow what is happening with that shield structure, we can see it's starting to move upwards, and it forms finally a narrow strip along the dorsal midline. The convergent extension of the hypoblast forms what's called as the chorda mesoderm. The convergent extension of the epiblast forms the neural keel, which is also will eventually develop into the neural tube. All these processes are happening during gastrulation. Now, Gastrulation is complete when epiboly is complete. So, epiboly is complete when the blastoderm covers the yolk cell by achieving 100% epiboly. And so, we see the entire yolk cell being covered. Now, the posterior caudal end of the embryo forms a swelling that's called as the tail bud. And that is what is shown in the figure J, where the arrow is pointing, which is the tail bud. So normally the moment the tail bud is formed, that is when gastrulation has come to an end because epiboly is complete. Following gastrulation, segmentation occurs. And this is where we start seeing different body parts form. Now we will not cover the process of segmentation in this specific set of slides. Just to show what we just talked about, here we can see the embryo that has come to the end of gastrulation, and the tail bud is shown where the arrowhead is, and they will have more developmental processes which involve segmentation to form the embryonic structure that is shown in the blue box. To summarize, we looked at different cell movements, the steps involved, as well as different structures that are formed during the process of gastrulation in fish.